Welcome to Inside Bioprocessing, a Thermo Fisher scientific podcast discussing key trends and topics within the bioprocessing industry. In this episode, we will be discussing the potential of next-generation cell culture media and feed systems to enhance monoclonal antibody production. Since their initial development in the 1980s, monoclonal antibodies, or mAbs, have become an increasingly popular biotherapeutic treatment option. And following the recent development of new therapeutic approaches based on mAb technology, this demand is only set to grow. However, supporting this increased growth is going to put significant pressure on mAb manufacturers to increase their production volumes without compromising on product quality or the economic feasibility of their process. To do this, manufacturers will have to increase their overall process productivity, and this will require the use of next-generation cell culture media and feed systems. To find out more about the need for high-performance media and feed systems during mAb manufacturing and how next-generation solutions could improve process productivity, we are joined by Chad Schwartz, Senior Global Product Manager at Thermo Fisher, and, speaking in a personal capacity, Michael Gillmeister, Principal Scientist at Regenex Bio. So, to start with them, Chad, could you give a brief overview of the current state of the CHO media and feeds market?
Yeah, sure. So manufacturers today, they really have two options when it comes to what their CHO cell lines will grow in. Either the pharmaceutical companies, they'll develop an in-house custom media, or they'll look to, one of the premier cell culture media suppliers for an off-the-shelf catalog offering. Of course, there are advantages and disadvantages to each approach. So it really depends on the preference of the company themselves. Factors, such as supply assurance, access to hydration capabilities, or the need for stability studies, are really at play and must be considered when taking either approach. As for the market itself, we've seen significant growth over the last decade or so, which really coincides directly with the growth of the pharmaceutical market. So as the pharmaceutical market grows, so does the CHO market in turn.
That's a good introduction, thank you. So why is choosing the optimal media so vital for mAb manufacturers specifically? Michael, would you like to talk a bit about this?
Yeah, so it's really important when we look at our manufacturing of antibodies and other products from mammalian cell culture to see what the consistency and reproducibility we get out from each one run to run. We need to be able to have predictable results from small scale PD all the way through GMP manufacturing. So one of the key inputs to that on the upstream side is the medium that the cells are growing in. So as the cells go from different phases, from inoculation all the way through production, be it in seed trains or shake flasks or production reactors, they're looking to see the cells are using the individual components of the media. So making sure that that media is optimized and designed correctly for the needs of those cells will really guarantee that we're going to get the productivity, titer, and product quality results we're looking for.
Great, yeah, that makes a lot of sense. What additional benefits do feeds provide, and how important is it that the media and feed combination used is complementary?
So as the cells are using up the components in the media and they're consuming and changing the concentrations of the media components, it's important that the feed that is then added back is reflective of what it started with for the individual basal medium. So not only is the feed necessary to be designed relative to the consumption of the cells, but also taking into account the other components in the basal medium and where it started with. So if you have a feed that isn't aligned or designed specifically for the basal medium, in addition to the cells, then you could end up with components that are over-concentrated or under-concentrated as compared to the starting concentration. Because not all the components in the medium are being consumed by the cells or necessarily being up or down regulated as the cell that your production is going on. So your feed is important to align with that and make sure it's complementing the overall production system.
Great, thanks for explaining that. So now we can go on to talking about the limitations of existing media and feed products. So Chad, what media and feed development challenges still need to be overcome?
It's a great question. Thank you for that. I think scalability is certainly important. Scientists like Michael in the pharmaceutical industry, they're tasked with developing processes, protocols at bench scale that also work at large scale in large bioreactors. However, due to resources and time, testing really only occurs at the bench. So it's important that the material that suppliers provide to these researchers can be seamlessly transferred to scale, to large scale. That's really both in terms of the manufacturability as well as the reproducibility. From a manufacturing point of view, it's important to consider format. Just for one particular example, most companies prefer media to come in powdered form. This helps to reduce costs of transportation and otherwise. However, the trade-off there is that the media must then be reconstituted at the site. So a media that will reconstitute easily and have little to no solubility issues during scale up is very important. Also, in terms of manufacturability, it's critical that the endpoint, those data points that these customers are looking for in terms of titer and otherwise, those results that are determined at bench scale also hold true at large scale. One does not want to obtain high titers on the bench, but then find out later that the performance does not translate at larger scale. Finally, the process needs to be reproducible. In this regard, elimination of potential hurdles or other variables is very important to pharma companies. From a technician standpoint, if one of the components has difficulties in reconstitution or tends to agglomerate throughout the manufacturing process, it's not lending to a reproducible process overall. I think, in today's world, these are just some of the items that still need to be overcome and are at the forefront of the minds of most pharmaceutical companies. Definitely interested, though, to hear some of Michael's thoughts on this topic as well.
Yeah, I think one of the interesting challenges that we've had recently in balancing media and feed development is looking at the combination or balance of the simplicity of use but flexibility. Chad mentioned reconstitution. So one of the things that we consider when we're looking at feed choices and feed design is for reconstitution into a single feed of how many components are going to go into it and how does that impact the need of our manufacturing. So trying to go from a PD lab or a research lab where we have the capabilities of making 1 liter or 5 liters of 10 liters with many different components is easier or more straightforward than going up to GMP manufacturing and to get multiple components into a several 100-liter batch where we're trying to balance out the speed and timing for that GMP batch. So when we look at feeds that have the option to have single components or even basal media for single component powders to go into its final formulation, it's a much more straightforward process for our GMP, both in the operator for the reconstitution, not having many steps, but also supply chain. So if we don't have to manage multiple part numbers or looking at different suppliers for some of the subcomponents, it makes it a much more straightforward and streamlined process. That it actually also works towards the actual use of the media in feeds. So if there's a system where we get really, really great titer or product quality, but it's like a 7 or 8 component feed system and requires different pH adjustments and a lot of operator interactions, that results in a lot of opportunities for risks where we could have a misfeed or they choose the wrong liquid when they're supposed to use part A versus part B, or if they miss a particular step, or just being able to make sure we have all the components ready at the right time. So the more complexity in the use is also a negative if it gets to the point where we have to balance out the benefit of choosing this component versus what is the actual implementation of using it in the large GMP scale.
Well, thank you both for your insights. I think that leads us nicely onto the next section, talking about essential elements of optimal media and feeds. So, what kind of capabilities will define next generation media and feeds? Chad, would you like to comment on that?
Yeah, sure. No, I think Michael did a really good job in the last question answering some of that. And anytime we talk about next generation, it really needs to be breakthrough. Pointing back to some of Michael's comments earlier, I think a next generation media and feed system need to solve those pain points that have already been discussed. It would need to be robust with high productivity in multiple cell lines. It would need to be, simple. To Michael's point, it needs to single part feed or single part media is critical to simplify the process and maximize the outputs. Further, it would need to be scalable, and have demonstrated results in a number of cell lines and processes. Further, in terms of regulatory concerns, it would need to satisfy a number of these needs, such as being chemically defined and animal origin free to help prevent variability in their processes. And finally, touched on it earlier, but and maybe most importantly here, to be defined truly as next generation, it has to have differentiated productivity, meaning that it lends to high titers, perhaps above 5 grams per liter, but also in turn promotes high protein quality. I think protein quality is sometimes overlooked during the process development stages. And a next generation cell culture media and feed system should promote the proper protein folding, the proper glycosylation profiles, and so forth that these pharmaceutical companies are looking for.
Okay, that makes a lot of sense. Is there anything that you'd like to add to that, Michael?
Yeah, and I think also being able to modify some of the choices of a platform, median feed to several different programs is important as well. So as we look at the impact on the medium and the feed on growth and titer and also product quality, we then want to implement that media and feed into program one or molecule two, and to be able to say, well, we only have to make small tweaks to the concentration or the timing of these different feed components to then be able to get to the product quality targets that we need would allow us to have a single supply, a single platform system, but then is applicable across several different molecules and hitting those individual targets.
That's a really interesting point. Are there any additional advantages suppliers themselves can provide to further enhance these products?
Yeah, I think one of the big challenges we've had over the past few years is been in supply, right? So that there's been a lot of supply challenges as we've moved through the science of the past few years, and there's been a lot of different ways to address that. So, for example, even before supply challenges, a lot of companies were trying to cover with redundant manufacturing, multiple sites, multiple different countries. But it's been a little bit of a challenge as well, even before that, when the redundant manufacturing isn't necessarily interchangeable. So I think being able to guarantee the supply with different sites is great if we're able to then carry forward the similar part numbers, for example. So if I order a part from site A, we being able to order the same exact part number with very similar raw materials is important from a different site, because that way we know that the quality of those individual raw materials that going in to make your medium and feed is going to be similar across wherever site it's able to actually be made at. Yeah, I think another thing that's really helpful for us as process development, for example, as we're trying to choose which medium to screen and how to understand the impacts of our individual system, is seeing data and examples that are really relevant. So, I'd say 5 or 10 years ago, a lot of the examples of medium development or feed development were using cell lines and example molecules or proteins or antibodies that were more from the academic side. So you were seeing titers and productivity levels that were not relevant or realistic to what we were making in industry. And I think it's been really helpful recently where a lot of the media suppliers have been able to get example proteins and antibodies and stable CHO systems that are making titers that are industrial relevant. And so that way, when we look at the data that's being presented at conferences, in technical papers by some of the media providers, we can actually get an idea of the productivities and performance of these different changes and how they might relate to our individual systems. But that being said, there's still a lot of variability that happens from cell line to cell line, from molecule to molecule. So the more examples that we can see of performance of a new product, a new medium, a new feed, and how it relates to not only the cell line that it was developed with, but also different customer cell lines, different example industrial cell lines, different molecules being produced, from antibodies to fusion proteins. It makes it a lot more applicable in understanding the flexibility of the media and the feeds and how they might apply to a novel system that we're going to be developing internally.
Thank you. So we're going to move on now to the final section. So talking about the future. So Chad, are there any emerging trends that you're looking out for that impact your cell culture performance?
Yeah, no, another really good question. And I'll give you a couple of examples. One's more related to performance, and the other would be, supplementary to the process. The first, as it relates to cell culture performance, a trend that we've seen quite a bit lately has been the reduction of raw material variability. And, when we look at these formulations, most cell culture formulations these days are made up of many different components and many unique components that are manufactured in a number of ways. And as through that manufacturing process, certain trace elements will be conferred upon or made up as part of those raw materials. Where we have seen the trends have been to try to reduce the presence of those trace elements or to reduce the variability of those trace elements in the raw materials themselves. So having techniques in place and quality systems in place to be able to monitor trace elements such as manganese and copper and otherwise that have been shown and have been demonstrated to affect cell culture performance is one trend that we are monitoring very closely and want to stay on top of in both the raw material standpoint as we receive raw materials in, and then on the finished good, on the actual media or feed itself. Another trend that I'll briefly mention is around electronic data, and that again is can be tied to performance. It certainly can, as we provide certificates of analysis and otherwise different types of documentation to pharmaceutical companies. They want to be able to consume this information electronically. And that is another trend that Thermo Fisher is getting out ahead of, is to be able to send documentations, upload them onto portals and otherwise that makes the pharmaceutical company's job quite a bit easier in terms of maintaining that data and having that data, at their fingertips. Those are the two biggest trends, that I've seen in cell culture that, are related to performance and are supplementary, maybe not always at the forefront, but certainly items that we are keeping at the top of mind as a supplier.
Sure, yeah. No, they both sound like quite interesting trends. Thank you. And finally, Michael, overall, what will enhanced productivity mean for both mAb manufacturers and other stakeholders within the industry in the long term?
I think the enhanced productivity, we talked about a little bit earlier about balancing how much additional titer we're getting and what the impact it is of the increased titer on product quality or predictability. So as the cells push out more and more of our product, of our antibody, then what is that doing to the post-translational modification, the different product quality attributes that we're looking at of the antibody? And at what point is titer enough and we are making what the product goals are as far as the productivity, and then are we then able to focus on other aspects of the antibody? So at that point, we can start looking at things like the predictability of the process. How can we ensure that it's scalable, looking from the PD all the way through our GMP manufacturing, and things other than just growth and productivity, which have been really important for the industry for many years. But at this point, we're really reaching some great productivity goals and levels that our systems can produce and really predictably reach. So there's a lot other aspects of the individual components of the media that are going into the impact of what our products and molecules look like. So I think that'll be an important further growth for the overall industry and media and feed development.
I think this is a great place for us to conclude our conversation. I'd like to thank both of our guests for their participation and insights. I'd also like to thank you, our listeners. If you'd like to learn more about the Gibco CHO Media and Feed Systems, please visit thermofisher.com/CHO Media or reach out to a Thermo Fisher representative.